Sugar is more than a source of sweetness. It's a molecule that fuels our cells, drives biochemical pathways, and profoundly influences cellular health. But how does sugar affect us at the molecular level? In this video, we'll take a deep dive into the cell biology and biochemistry of sugar metabolism. Sugars are carbohydrates classified as monosaccharides, disaccharides, or polysaccharides. Glucose, the primary monosaccharide, is an essential energy source for cells. Sucrose or table sugar is a disaccharide composed of glucose and fructose, while lactose is a combination of glucose and galactose. Each sugar has a unique metabolic pathway and the role in the body. Sugar digestion begins in the small intestine. Glucose and galactose molecules are absorbed through the SGLT1 transporter, which uses sodium as a co-transport mechanism. Fructose enters the enterocytes via the GLUT5 transporter, a sodium independent process. Once inside the cells, these sugars are transported into the bloodstream through GLUT2 transporter, where they travel to the liver for metabolism. After the absorption, glucose is taken up by cells through tissue-specific glucose transporters or GLUTs. For example, GLUT1 ensures basal glucose uptake in most of the tissues, while GLUT2 handles high-capacity glucose transport in the liver and pancreas. There are other transporters such as GLUT4, which are found in muscles and adipose tissues, is regulated by insulin. Insulin is a hormone secreted by pancreatic beta cells in response to high blood glucose, binds to its receptor and activates a cascade involving IRS1, PI3K and AKT, promoting the translocation of GLUT4 to the plasma membrane. Once inside the cells, glucose are involved in the glycolysis, which is a 10-step process. This process converts one molecule of glucose into two molecules of pyruvate. Key regulatory steps includes hexokinase or glucokinase enzyme, which traps glucose in the cell by converting into glucose 6-phosphate. Second one would be phosphofructokinase 1. This is a rate limiting enzyme activated by AMP and inhibited by ATP and citrate. Pyruvate kinase generates ATP and forms pyruvate, which can enter the mitochondria for further oxidation. In the mitochondria, pyruvate is further oxidized into acetyl CoA by pyruvate dehydrogenase, entering the Krebs cycle in the mitochondrial matrix. This cycle generates 3 NADH molecules, 1 FADH2, 1 GTP per turn, which donates electrons to the electron transport chain or ETC. The ETC generates a proton gradient driving ATP synthesis through ATP synthase, producing up to 32 ATP per glucose molecule. That are 32 ATP molecules per one glucose molecule. When glucose intake exceeds energy needs, it is stored as glycogen in the liver and muscles via glycogenesis. Glyco is glucose and genesis is generation. Excess glucose is converted to fatty acids through de novo lipogenesis, involving enzymes like acetyl CoA carboxylase and fatty acid synthase. This leads to triglyceride accumulation in adipose tissues and in some cases non-alcoholic fatty acid or fatty liver disease. Chronic high sugar intake downregulates insulin receptors, disrupting the IRS1, PI3K, AKT pathway. This leads to insulin resistance, characterized by elevated blood glucose and increased pancreatic insulin secretion. Excess sugar overloads mitochondrial pathways as well, increasing the production of reactive oxygen species or ROS. This ROS damages DNA, proteins, lipids and triggers chronic inflammation through the activation of natural factor kappa beta. Glucose contributes to protein glycosylation via the hexoamine biosynthetic pathway. Overactive glycosylation modifies proteins involved in insulin signaling, further exacerbating metabolic dysregulation. High glucose level leads to non-enzymatic glycation of proteins, forming advanced glycation end products or AGEs. These AGEs interact with RAGE receptors, triggering oxidative stress, inflammation, and which eventually contributes to aging and diabetic complications. Hyperglycemia activates apoptotic pathways in pancreatic beta cells and endothelial cells through increased ROS production and activation of stress kinases such as JNK and P38 MAP kinase. So, sugar is a powerful molecule. It sustains life but can also disrupt it when consumed in excess. In our next video, we'll explore sugar's impact on the brain, focusing on addiction, cognition, and neurodegeneration.